This video tutorial is all about population growth in a closed culture. I'm going to run through serial dilutions, the growth curve, and then towards the end just mention sort of the primary and secondary metabolites that we mentioned before with reference to fermenters. A liquid broth can be used to measure the growth rate of a microorganism population. A sterile broth is inoculated and the population size measured at regular intervals during incubation. The population size can be measured by transferring a small sample to an agar plate and incubating the agar plate. Each individual organism will produce a visible colony. The number of individual microorganisms in a broth can be very high. If the broth is inoculated on an agar plate, there may be too many colonies which merge together, making a count impossible. In order to investigate the rate of growth of the population of microorganisms, it will be essential to reduce the population density, and this can be achieved through serial dilutions. Now, serial dilutions is just a stepwise dilution of a broth culture. At each step, the broth is diluted by a factor of 10. So you take 1 centimetre cubed of the sample of the broth and add 9 centimetre cubed of distilled water. You then label the diluted broth as kind of times 10 to the minus 1. And then from that, you take 1 centimetre cubed of this diluted broth and add 9 centimetre cubed of distilled water, and you can label that times 10 to the minus 2. And you continue this procedure until you have a series of dilutions which are sort of suitably labelled. A drop of each dilution can be used to inoculate an agar plate. One of them will produce a culture plate in which the number of colonies can be counted. When recording the population density, you just shouldn't forget to multiply your count by the dilution factor and also by the volume added to the plate. We're now going to look at the growth curve. A small population of microorganisms in a closed culture that contains all the nutrients required for growth will undergo population growth. A closed culture refers to a population in which all the conditions are set at the start and there is no exchange with the external environment. The population growth will follow a predictable pattern, as shown on the slide in front of you. These are similar to the conditions set up for batch production in a fermenter. In batch production, however, certain substances such as oxygen may be added to keep the population growth until the nutrients are all used up. So we're just going to go through this growth curve step by step. Firstly, the lag phase. In the early part of the population growth, the population does not grow very quickly. This is partly because the population is still small, but also because the organisms are adjusting to their new environments. This may involve taking up water, growing the cells, switching on, activating certain genes, and synthesizing specific proteins, enzymes. Next, there's the log or exponential phase. In the log or exponential phase, the organisms have already adjusted to their environment. They each have the enzymes needed to survive. Each individual has sufficient nutrients and space to grow rapidly and reproduce. The population doubles in size with each generation. In some microorganisms, this can be as frequent as once every 20 to 30 minutes. Next, we have the stationary phase. So eventually, the increasing numbers of organisms use up the nutrients and produce increasing amounts of waste products such as carbon dioxide and other metabolites. The rate of population growth will decline and the number of individuals dying will increase until the reproduction rate equals the death rate. Now, this is the stationary phase because there's no population growth. Finally, we enter the death or decline phase. The nutrients run out and the concentration of waste products becomes lethal. More individuals die than are produced and the population begins to fall. Eventually, all the organisms will die. Finally, I'm just going to mention the primary and secondary metabolites. Primary metabolites produced during the normal activities of microorganisms will be collected from a fermenter during the log phase. In a fermenter, the population is not kept in a closed culture, but conditions are maintained for optimum growth. Secondary metabolites are produced in the stationary phase. The population must be kept in a closed culture, and the metabolites can be collected at the end of the stationary phase or during the decline phase.